Good afternoon. First of all, I would like to thank to my supervisors the opportunity to talk this afternoon about the pilot study we ran in Lanzarote. And I'm going to, first of all, explain the idea of why we ran this study, because we already have some studies, as Tommy Johnson have explained before. So why did we do another study, and why did we design it in the way we did it? So that's the goal of this talk. So just to summarize the result of the previous studies, I'm talking about Stefan's study and Tommy's study. There was a greater reduction in waist circumference significantly compared to the control diet. And also weight loss in Tommy's study was also, weight loss was also significantly higher in his study. If we look at the macronutrient composition in Stefan's study, there was a significant difference in the amount of carbohydrates. In this case, it was lower than the Mediterranean diet. With regard to protein, it was significantly higher compared to the Mediterranean diet, and no difference in the intake of fat. And here you can see there was no difference in the intake of alcohol, most of it to make the meals. And there was also no significant difference in the amount of fiber. When we look at the macronutrient composition in Tommy's study, we see more or less the same. So this is the amount of carbohydrates. Carbohydrates was significantly less in the Paleolithic diet compared to the diabetes diet. The amount of proteins was higher again. And in this case, the amount of fat was significantly higher than the diabetes diet. In this case, there was a significant difference in the amount of fiber, perhaps because of the lower intake of carbohydrates, and no difference in alcohol intake. Then another factor is glycemic load. So there was a significant difference in both trials, where the glycemic load was lower in the Paleolithic diet compared to the control diet. So we will come back to this later on, and I will explain you why I presented this data from their studies. Now I'm going to try to explain the physiopathology in type 2 diabetes, or some ideas. When I dig into the literature and try to understand what is the cause or what are the mechanisms that can lead to type 2 diabetes, is often often I read about insulin resistance and a decreased function in the pancreas. So this means a decreased production of insulin when the patients eat, especially carbohydrate meals. And it's, for me, it's striking and surprising not to read about glucagon. So I think glucagon is most of the times forgiven and taken out of the equation when we talk about the development of type 2 diabetes. And it's also surprising that even though insulin and glucagon were discovered more or less at the same times, in the early 20s, glucagon has received much more attention in the medical community. So we try to focus more in insulin rather than in glucagon, especially after the discovery that we can treat type 1 diabetes with insulin. So what is the role of glucagon and insulin in physiology and in type 2 diabetes? The main role of insulin in diabetes is to control the production of glucagon. So when glucose raises in the blood, that makes the beta cells produce insulin, and the concentration of insulin that reach to the alpha cells is higher than in any other part of the body. And this raise of this communication between the beta cell and the alpha cells is mainly to control or decrease the production of glucagon. And this mechanism seems to be disrupted in diabetes. In type 2 diabetes is disrupted, and of course in type 1 diabetes we don't produce insulin at all. So glucagon is like, it doesn't have a break. It doesn't have, it's the hormone that controls its production. And Roger Unger, he has many publications about the role or the prominent role of glucagon in type 1 and type 2 diabetes. He published a very nice paper in 2012 with title is Glucagonocentric Restructuring of Diabetes. So what we see in patients with diabetes is that after a carbohydrate meal, glucose goes up, and at the same time insulin raises, although not as much as it should increase. But if we look at glucagon, we see a paradoxical increase in glucagon. In physiology, glucagon should decrease after eating a carbohydrate meal. So this is a paradoxical effect of glucagon. And what does this mean? It means that after eating a carbohydrate meal and after digesting carbohydrate, they become into glucose, and glucose raises in the blood. And at the same time, if you are not able to decrease the production of glucagon, you're going to produce endogenous glucose from your liver. So this is like a double hit in the levels of glucose in blood. And when I read this paper, I got hooked with this information because I think it's forgiven in the medical literature, and I think it's quite important. And for example, we know from animal studies and some human studies that if glucose increases and we don't produce insulin, you don't have a decrease in glucagon. Glucagon goes up. And this effect is paradoxical. So we need insulin to decrease or to control the production of glucagon. And if you don't have insulin, then glucagon, the fasting levels or the postprandial levels are higher than they should be. As an example, I'm going to show you this interesting experiment where they tested the production of glucagon after a 50-gram oral glucose tolerance test and an isoglycemic intravenous glucose infusion. There were 10 patients with type 2 diabetes. You can see them, the results here in dark green, and 10 healthy people. So in the baseline, you can see a significant difference in the levels of glucagon between healthy people and patients with type 2 diabetes. So the baseline levels of glucagon are higher in patients with type 2 diabetes. So here in the open circles, we can see the levels of glucagon after the isoglycemic. Well, just to explain it, what they did was first the oral glucose tolerance test with 50 grams of glucose, and then they did an isoglycemic intravenous glucose infusion, and they copied the curve. So the curve after the oral glucose and after the intravenous glucose was exactly the same. But if we look at the levels of glucagon, in healthy people, after the glucose infusion or after ingesting glucose, you can see a decrease in the production of glucagon. And you see the same when you give the intravenous glucose infusion in patients with type 2 diabetes. We see a decrease in glucagon, which is physiological. But when you give the glucose, the oral glucose tolerance, you see an increase in glucagon, and this increase is paradoxical. So this seems to be the hallmark of patients with type 2 diabetes, hyperglucagonemia. So fasting increase fasting glucagon levels and an attenuated decrease in postprandial glucagon. If we look at some studies in humans, in this case 26 healthy people, they ate 1 gram of carbohydrate per kilogram of body weight in different forms. So in different days they ingested glucose or white bread or 2 kinds of whole wheat bread. One was standard whole wheat, and in the other day it was ultra fine whole wheat bread. And what we can see is that after the oral glucose ingestion, a physiological decrease in the production of glucagon, which makes sense. But after ingesting the same amount of carbohydrates, but in this case in form of white bread or whole wheat or ultrafine whole wheat bread, we see an increase in the production of glucagon. And this again is paradoxical. And we know from studies in dogs that some peptides derived from the enzymatic digestion of wheat, they can bind opioid receptors, and this binding of these peptides, they are called exorphins A5 and exorphin B5, they induce an increase in glucagon. So there seems to be an interaction between some peptides found in food and opioid receptors, affecting the endocrine system. And this is one of the questions that we are trying to answer with our studies. How does food choice affect the endocrine system, independently of weight loss, macronutrient composition, glycemic load, or physical exercise? So we are really interested in what's going on in those receptors, in those endocrine receptors, and the response to key hormones like glucagon. But we will come back to this later. Another hormone that seems to be very important, and Tommy has talked about it before, is leptin. So let's review a little bit the roles or the functions of leptin in human physiology. Well, leptin, as we already know, is able to control appetite through the brain. And also, leptin is a key hormone in lipid partitioning. So what does lipid partitioning mean? It means that when we are faced to a positive energy balance, we must store the excess of energy somewhere. And the tissue made for this purpose is the adipose tissue, of course. So when we increase the amount of, or we are faced with a positive energy balance, we should store the energy in the adipose tissue. In this case, the levels of leptin goes up, and very interestingly, the adipose tissue becomes leptin resistant. But tissues like the pancreas, heart, muscle, and liver, they should be leptin sensitive. Why? Because leptin increases the activity of the mitochondria to burn that fat. So leptin is a key hormone to avoid ectopic fat deposition. So it's not only important the amount of fat, and Tommy has already talked about this, in your body, but it's important where do you store the fat. And it looks like a fatty liver and a fatty pancreas are key characteristics of type 2 diabetes, which means insulin resistance in the liver and pancreas failure when you have ectopic accumulation of fat in the liver, in the pancreas and liver, and also in the muscle that could lead to insulin resistance. And also from animal studies, we know that leptin is able to control the production of glucagon through, well, it's not clear how this works, but there seems to be a direct communication and also a pathway through the brain. So animal models for type 1 diabetes, if you inject them leptin, you can prevent the development of diabetes. So leptin seems to protect against diabetes. And also from before, there are animal studies in mice also showing that mice with type 1 diabetes, this means without beta cells, if these animals are knocked out for the glucagon receptor, they don't develop diabetes. So the action of glucagon seems to be more important than the role of insulin in diabetes. Okay, and also we know that leptin improves the function of the islets in the pancreas. The question is, what can affect leptin sensitivity? There are many factors. One of them could be inflammation. There are some studies suggesting that chronic low-grade inflammation can induce leptin resistance. But we know also from some studies that some bioactive compounds, and Stefan already talked about these substances in some plants, especially in seeds, they can potentially bind endocrine receptors such as the leptin receptor or the insulin receptor. For example, lectins. Lectins are glycoproteins found in seeds and legumes, and some studies suggest that they can bind key receptors, as I said, like the insulin and leptin receptor. And in this study, they showed that when you increase the concentration of wheat germ agglutinin, which is found in wheat, the binding of leptin to its receptor decreases. And Tommy brought a very nice review paper about this topic that you can read if you are interested in agrarian diet and disease of affluence. So, do evolutionary novel dietary lectins cause leptin resistance? That's one of our interests in our studies. So we are, or at least we are trying to answer these questions with more studies. And from Tommy's study, this data has not been published yet. There was a trend towards the decreased glucagon after the Paleolithic diet, and also a trend towards decreased leptin in the Paleolithic diet. So to put the whole story together, it seems like food choice and evolutionary food choice can improve key hormones like glucagon and leptin. So that's our working hypothesis, and this data was very important in order to design the trial that I'm going to show you now. So as a conclusion, as a summary of this first part, glucagon and leptin are key players in type 2 diabetes, and bioactive compounds in cereal grains and legumes potentially can disrupt the function of these hormones. So now I'm going to talk about this short, very small pilot study we conducted in Lanzarote. You know this island? If not, I highly recommend you to visit it whenever you can. So I would also like to give thanks to our sponsors. This is the main institution in the island, Cabildo Lanzarote, Town Hall of my town, and Arrecife Town Hall, which sponsored the study. I would also like to thank my supervisors and collaborators and friends who have made it possible to do this, although very short and small trial, but a lot of work, a lot of hands-on work and logistics, but at the end we did it. Okay. So we recruited at the beginning 15 patients with type 2 diabetes, although the first day we had to exclude one participant because of lack of compliance at all. He didn't show up even in the first meal. And increased waist circumference. The cutoff line was 80 centimeters for women and 94 centimeters for men. And they could be on either hypoglycemic drugs or insulin. The intervention consists of a crossover Raman-Oda design. There were 4 + 4 weeks, separated by 6 weeks washout period. So the patients either started with a Paleolithic diet or the diabetes diet according to the Spanish recommendations for patients with type 2 diabetes. And we will see more details later on. We performed lab tests in the fasting state, and after 15, 30, 60, 90, and 120 minutes of 75 grams oral glucose tolerance tests, and we also perform anthropometric measurements at baseline and end of each intervention. So the patients ate 2 healthy diets with or without cereal grains, legumes, or dairy products. So this was the dietary design. And according to Spanish dietary recommendation for patients with type 2 diabetes, we met the requirements for macronutrient composition, minerals, vitamins, and fiber. I will come into more details now. We gave the patients specific instructions. So I gave a 7-day menu plan, and they were encouraged to eat as we told them in those instructions. Sorry. And one of the main goals of the study was to avoid weight loss. So because in the previous studies there was weight loss and also decreased waist circumference, we wanted to control those confounders, or potential confounders. So we want the patients to be weight stable, and we elaborated different versions of this diet. So we had the starting point at 2,000 calories, and now you will see the macronutrient composition. And then we made different versions, 1,800 calories, 1,600 calories, 2,200, 2,500, and so on. So every week we weighed the patients, and if the patient changed its weight more than 1 kilo, we readjusted the diet so that we kept them weight stable. And we did. We succeed with the weight stability. So at the end of the 4 weeks in the 2 groups, in the 2 diets, the patients didn't lose weight and they didn't decrease waist circumference. Okay. So this is the macronutrient composition. Also, I would like to go back. Yes, when we elaborated the meals, we use a validated dietary program that can classify the diets as healthy, very healthy, poor diets, or inadequate diets. So it turns out that with this program, the Paleolithic diet was very healthy. So it was very healthy, so no negative points to make about the diet. And this is a macronutrient composition, the same amount of carbohydrate, the same amount of protein, and the same amount of fat. So we design two diets with the same macronutrient composition, but with different food choice. And as I said before, in one diet, there were no grains, no dairy products, and of course no sugars, and no refined oils. Glycemic load was also the same, what was one of the differences in the previous trials. So we wanted to adjust this confounder or potential confounder. And very interestingly, when we designed the diet, we found that the amount of fiber was very high. So in order to reach a percentage of 52% of energy from carbohydrates, you have to include a big amount of fruits and vegetables and tubers, and you will see the figures right now. So they ate almost 1 kilo of fruit every day. And when the dietitian gave the advice to the patients, the patient said, what? Do we have to eat all this amount of fruit and all these bananas? They ate bananas every day, like 2 bananas on average. And they were, this is going to be awful for my blood sugar, for sure. Okay, let's see. And it's also very important that when the dietitian gave the recommendations, he said, or she said, that we didn't know which diet was the best, if any. So in order to avoid any kind of subjective emphasis on one diet or the other, we make it clear that we didn't know if one diet was better than the other, if any. So the patient didn't know anything about Paleolithic diet or the evolutionary template. They knew nothing. And I even didn't mention the name Paleolithic diet, and I termed healthy diet A and healthy diet B. That's it. So in that sense, they were blinded to if one diet was better than the other, which is a very common problem, as Stefan said, in many nutritional trials. If you encourage the person to eat a diet and the other group is a control group, then the motivation is not the same. And also it's very interesting that we did a lab test for the inclusion criteria, and then we did again lab tests in the baseline. So all the patients improved from the inclusion criteria lab test until 4 weeks later with the baseline lab test. So this means that every time that the patient starts a diet or is followed by someone, or he has to fill questionnaires about food, etc., he automatically improves his diet. So, I mean, compared to the average food pattern in the world, anything is better. So I think it was really important to not to state our beliefs about the diet in this trial. Okay, so here you can see, we published the protocol. It's published in the journal Trials, so you can see all the details, all the nutrient composition, how we gave the recommendations, and so on. Of course, we see a difference between both diets with regard to cereal grains, legumes, and dairy products. And also here you see that the amount of vegetables in the Paleolithic diet was significantly higher. So in grams is 420 grams. Fruits, almost 1 kilo. Sorry, this is energy content. Sorry, it's not weight, it's energy. So almost half of the energy comes from fruits. And here you can see 420 kilocalories out of 2,000, which is very high. The amount of meat was also significantly higher, and the amount of oils lower in the Paleolithic diet. This is the food choice for this diet, as you knew from before. And if we look at the micronutrients, there was no deficiency in anything except, for example, if you look at vitamin D, and of course, if you exclude dairy product, the intake of vitamin D would be lower than the Institute of Medicine recommendations. So in this graph you can see in purple 100% of the recommendation of the Institute of Medicine, and in green the amount achieved with this diet. And also we can argue about calcium, because if you exclude again dairy products, the intake of calcium would be lower, but it's another story if this is unhealthy or not. So no evident nutrient deficiencies. And the patients ate lunch in the hospital, so they was more or less very controlled, the trial, because they went every day. They met the dietitian every week. We weighted them every week. We found them also every week, so they had contact twice per week with the investigators. So I think it's pretty good in 4 weeks. So that means 8 times of contact in 4 weeks in each diet. And we also asked them if they noticed any kind of difference when we gave the recommendations. If they notice more enthusiasm with one diet or the other, and they say, no, we felt that the investigator gave the same amount of kind of information with both diets. So we think that we actually tested the effect of food and not other confounders. So we gave the food in this trolley, in these trays, marked with B or A, and then in the lunch room we separated in 2 with the label A and B. So this is how we organized the lunch room. So here you can see a picture of some of the meals. And the main outcomes, the primary outcomes are fructosamine. Of course, we cannot use A1C because it's only 4 weeks, so we will use fructosamine. And fasting glucagons, we are really interested in glucagon, and many other variables. We only have some preliminary results from the lab in Lanzarote, so most of the variables are yet to be tested. We haven't analyzed them, but we have fasting glucose, and there was no change compared to the baseline during the diabetes diet, but there was a significant drop within the Paleolithic diet compared to baseline, although the difference between diet was not significant. But it's interesting that in 4 weeks, without weight loss, and also we told the patients to do the same amount of exercise in both diets. So if they walk for 30 minutes every day, to keep this amount of exercise in both diets. So it's interesting that we overfed them. We had problems to keep them weight stable because they tend to lose weight, and we had to overfeed them. So it was very challenging to keep them weight stable, but we did it at the end. And even though we overfed them, that they didn't change exercise, well, this is interesting, although there was no difference between the diets. And there was a trend towards a difference between diets in HDL. So in the diabetes diet, HDL decreased, and there was no change in the Paleolithic diet. So as a conclusion, so glucagon and leptin seems to be key players in type 2 diabetes. Evolutionary food choice might have an effect on the endocrine system independently of weight loss, macronutrient composition, glycemic load, or fiber. Promising data from a study based on, again, evolutionary food choice, and there's an urgent need for more studies, long-term study.
Thank you very much. There will now be coffee served and fruit. If you want cookies, I think you can buy them in the over the counter. I think there is 2 fruits for everyone, because we thought maybe 30 people will come. So I'll just try to find it, and we'll gather at 3:20.